Hi, so my talk today is about lizards' physiology and how they can cope with climate change. How can they or how can't, you will see. I will use some examples of not only lizards, but also ectotherms. So for the start, let us just look at this very basic background that we need to know when we are talking about ectotherms. So what is ectothermy? That is a trait of organisms that don't have any internal processes to produce body heat or to maintain stable body heat. And they are therefore having also a very variable set of body temperatures, which makes them also poikilothermic. This is another expression we use for the organisms that have a variable body temperatures. And amongst others, they are not only lizards, of course. Ectotherms are a huge amount of species that occupy our planet, all the invertebrates, and amongst vertebrates, there are amphibians and reptiles, and they can occupy different habitats. We may think that because their body temperature is dependent more from the environmental temperatures, that they will not be maybe able to survive in either too hot places or too cold places. But actually, this is not the case. If we look at this map of the world distribution of, for example, ant species, we see they occupy all continents except really the far north or the south pole, but they are occupying really a large set of environmental temperatures, also cold and hot regions. The same is true for example, reptiles, where that also encompass lizards. They are present in also hot, cold, and really diverse temperature range of environment. So how do they do it? Yes, that is the question, and that's something I want to start with my talk, even though I will focus later more on our research done on the metabolism. But we need to really, because temperature and metabolism are linked, we need to know how the organisms, ectotherms, are obtaining and maintaining their body temperature, or is this temperature varying according to the environment. So this would be what would happen if, for example, an ectotherm organism would not thermoregulate. So that would mean when we will go outside and catch, for example, this organism around 20 degrees of environmental temperature, we would measure the body temperature, and it would be also similar, around 20. And we do that across the temperature span and get these patterns. So these patterns are common for the thermoconformer organisms. Those organisms don't use some sort of thermoregulation, and their body temperature is actually reflecting the environmental temperature. But this is not the case in lizards, we know that. So when we go outside and we catch a lizard and it's 18 degrees, for example, we measure the body temperature and it's gonna be maybe around 35, like it was in this study of this species of lizards. And then we do that across the day or across the temperature range, and we get this pattern. So body temperatures are always around 32 to 36, so there is some sort of let's say some sort of range of body temperatures in which these lizards are able to thermoregulate to along quite a large span of environmental temperatures. How do they do it? They are using thermoregulation. And I think this comics of Gary Larson really well describes how this process is going on in lizards. And it's like this. They move from hot or warm environments to cold environments. And they do that constantly. They do that across the day and very precisely. So they are very good at it. they will constantly and really determinedly move between these cold and hot places to obtain and maintain their body temperatures in the range that is actually their optimum or preferred body temperature, as we call it. We can also investigate this. So when we want to know what is this preferred body temperature range in lizards, we usually put them in this very long terraria where we make a gradient of temperatures. On one side we have usually 40 degrees, on the other side 20 degrees. Then we let this lizard to move around and thermoregulate, and we measure their body temperatures. For example, in this Zootoca vivipara, in the viviparous lizard, we measured it from 9 in the morning to 18 in the afternoon every hour, and then we get these results. Here I show you an example where I want to show that males and females, for example, they had a slightly different range of preferred body temperatures. This is very interesting because in this season, when we did this study, the females were gravid, which means they had eggs in their body, and they are also in that time of the year when they're gravid and they have eggs that are developing inside their body. They would usually, females, select for a lower temperature set point. And that is also interesting because then we know that the selected preferred body temperatures can also be different. For example, here, if the status of the animal is different, and we also know between seasons it can be different. So this is also a very important thing we need to know when we are gonna, you will see when we're gonna think about metabolism and the rate of metabolism. And why is this temperature really, really important for them? Why do they need to achieve it? Of course, in this set of temperatures, in this set point, you can see there are different performances. For example, in this study, they measured sprint speed, they measured endurance with the green, and they measured also the energy assimilation. So everything is peaking at this range of temperatures. Why? Because also metabolism is, of course, temperature dependent. The enzymes have a certain optimum temperature range, or even more, it's even narrower, as you see, than the other performance curves. This is a very narrow range of temperatures where the enzymes are optimally functioning, and that is when the metabolism is the most efficient and most highest. And this is also the same as with other, it is optimizing the other performances of the lizards. So achieving these temperatures and maintaining them in a narrow range is, for an ectotherm that is thermoregulating, very important. But not only temperature, I have to mention here also another limiting, or let's say another factor in the environment that is very important also for, of course, for the metabolism. So this is water. And in lizards, we nowadays know quite a lot. So we more or less know that the species can differ in how much water they can lose, if, for example, during desiccation. And how do they do it? Well, the water is actually, they lose the water from surface area through cutaneous evaporation. Some of the part of the water can be lost by respiratory evaporation, and then, of course, by secretion. And how do they gain it? Mostly from food and sometimes also from drinking. So the water in metabolism is a key component. Without it, the metabolic pathways, biochemical reactions cannot occur. So we need to know this is very important. And as I said, it's already known for lizards that even, for example, this is in Tarantula maritanica, a study done by colleagues from the Functional Biodiversity group. They measured water loss rates in a desiccation experiment between different lineages of Tarantula maritanica. And even between lineages inside the species, we see there are differences between species. So species do have some certain specific way of handling water losses. And even more, we did in 2019, we were involved in a really large-scale study where we looked at phylogenies of the Lacertids, of this big group of lizards that live in Europe, Africa, and Asia. And here on the left, there is the warmer temperature is represented with warmer colors, and the colder temperatures of the environment are represented with this colder tone of colors. And we plotted this against in the phylogeny. But not only that, we also had the water loss rates for a lot of species, so we could compare these two. So from which environments are coming, in which environments species are losing more water or less water. And when we looked at this relationship, you can see this is the hotter the place, the more hours of the year with above 30 degrees, or the more water they lose, right? So we have this negative relationship, meaning that species that occupy very hot areas, they lose less water, whereas the ones that live in more colder places and more humid places, also we've identified those places are commonly also more humid, they don't lose so much, they lose a lot of water, they have higher water loss rates, right? And knowing that, also together with other results of this extensive work that was looking at evolutionary rates, the main conclusion was that we found out that the lizards that live today in cold and also humid areas are the ones that might suffer due to global warming in the future the most. And this is since we know that the climate is warming per se, so the temperature is rising, but we also know this is bringing also droughts to some humid places, so they are drying up, and also there are these cases of desertifications happening in some places, and in those places where lizards are mostly having huge or let's say high water loss rates, they will suffer there. And that is also important to think about when we are talking about climate change. But now let's go to the topic that I want to today give you some examples of. This is how actually these responses, what do we know about that? Are they able to respond also on the physiological part? So I'm not going to talk about behavior, how they could behaviorally respond to some sort of global warming or desertifications or other climate change effects, but I will talk mainly about metabolism. Is there any knowledge about whether there is a sign of adaptation or plasticity in metabolism that will tell us that they will be able to respond to some rapid changes? First example that I want to talk about is the two species that I've worked with during my PhD. This is a Horvat's rock lizard, Lacertid horvati. That's an endemic species that lives in the southern Alps and the northern Balkans. And the second species is the Commonwealth lizard, Podarcis muralis, and it's a widespread species. And during my PhD, I was mainly answering myself this question: what would happen under climate change with these two species? And why with these two species? Well, it is very interesting. Their distributional pattern is very interesting. The Commonwealth lizard is more common in the lowlands, but it can also occupy here until 1,000 in my study area. And on the other hand, the Horvati has the opposite pattern, and they have this high overlap. And then I was thinking this lowland species probably is more adapted to warmer climate. So with climate change and global warming, it might push to be more abundant also in the higher elevations. And what would happen? What is going to happen with the Horvat's rock lizard? Is it going to be pushed to go further up, or maybe they can coexist or not? And if it's going to be pushed up, it might be a problem for them if there is no higher elevations available in the, for example, the study area. So I had this idea of competition behind, because the two species are really, really similar. When you look at them in the field, you have to be quite an expert to really distinguish them. Just by seeing them, they are super similar. And that's why I was questioning whether they are in competition, and is this competition going to be somehow influenced by climate change as well. And as a first step, I was really interested in their temperature. So I told you, I was thinking one of them is more adapted to warm, one of them is more adapted to cold. So then I was expecting maybe to find some different temperature set points, these preferred body temperatures. I did the experiments with both. And this is the results we get, which were really interesting. So what we found when we compared the spring and summer preferred body temperatures, you see here in the high altitude species, Horvati, we found some differences. So they were a bit lower in spring and a bit higher in summer. But what is even more interesting is that in the lowland species, in the Commonwealth Lizard, we've seen this huge, and it's a significantly larger difference in the temperature set points between spring and summer. So they thermoregulate to a lower set point in spring and to quite higher, whereas Horvati is always trying to achieve a more similar set point, and being in spring, when it's actually quite cold outside, can thermoregulate to a higher set point. This is kind of showing that the high altitude species is behaviorally specializing to achieve a narrower set point and also higher temperature, preferred body temperatures in the cooler time of the year. And just by chance, in the group of National Institute of Biology in Slovenia, where I had also a supervisor, they are mostly working with invertebrates, but they are using these two techniques. They are using respirometry to estimate the metabolic rates, and then they also use a biochemical reaction assay test that is determining the metabolic activity. And that is actually, it's measuring an amount of oxygen reduced in metabolic enzymatic reactions. And this is giving us this idea of this intrinsic tissue level metabolic potential for that enzymatic capacity for metabolism. And these two techniques were available for me during my PhD. So I also just by chance said, okay, let's see, maybe we find some interesting, or we find some differences or similarities between these two species on the metabolism level. And this is where my path with metabolism studies started, actually. And here are the results. So when we compared their respiration rates, so the amount of oxygen they consume when we measured their breathing, we did that around their preferred body temperature of both species, and then also a bit lower, just to see the effect of temperature. There were no differences between the species. But when we did this test of the biochemical assay, so what is the enzymatic capacity that they have at the tissue level, then we found differences. And this was really interesting. So we found there is already some fundamental, really baseline difference on the enzymatic level of mitochondria, if you want me to say like this, in both species. The high altitude species has a higher enzymatic capacity in the tissues than does the Commonwealth lizard. And what could that mean? Well, now here I want to stress it's really important to take into account to which temperature they are thermoregulating, because the metabolic reactions, they're directly linked to temperature. So if a lizard has a higher body temperature, their metabolism is higher. But knowing also, for example, that Horvati, the high altitude species, had a higher temperature set point, was achieving higher temperature set points, and it was also more precise thermoregulator. And on top of that, had also a biochemical adaptation in the metabolism to have a higher enzymatic capacity. So all this together implies that this is some sort of adaptation on the metabolism level together with behavioral thermoregulation, to optimize it for maybe, let's say, harsh and cool environments. So we know they are distributed more in the mountains, and apparently this is some sort of adaptation. On higher up in higher altitudes, there are lower temperatures generally. Average environmental temperatures are lower, and then also lizards will come out later in the spring, and they will retreat to refuges and to the overwintering sooner in autumn. So they have this shorter activity period in the season than do the animals that live lowland, where it's warmer, and they can come out sooner and they can retreat later. So it's very important. But we found these couple of things. So also the behavioral thermoregulation and metabolism being optimizing for a, let's say, a rather cooler and shorter activity periods in the high altitude species. Seeing that, we then got an idea whether we could test this maybe in also other ectotherms. So we decided to do that. We found species pairs that had really a similar distribution, and these candidate species were our two lizard species that I talked to, and we had two species of salamanders, alpine salamander and the common salamander. Also, one is narrowly distributed, the other broadly, and one is high altitude, the other is low altitude, and the same with two species of ground beetles. And we looked at metabolism in these three species pairs. A very important point here is that it was very important how we sampled. So we always sampled in this middle altitude. So the high altitude species is distributed in high altitude, but also goes down. And then there is a low altitude species that always also is distributed in lowlands, but then it's also distributed a bit up. And there is this band of middle altitudes where both occur. And why did we sample here? It's important because we didn't want to detect some sort of local adaptation. If we would sample a high altitude species from the peak and the low altitude species from the bottom, we would clearly see maybe some local adaptation to that conditions. No, we wanted to sample them under the same environmental conditions to see if they have this intrinsic adaptational sign in the metabolism. So we sampled them at the same altitude. And when we looked at their metabolism, it was really cool. The result was really cool. We actually got confirmation that the high altitude species, here is in lizards, in the middle is in salamanders, and in the right is with ground beetles. All the time, the high altitude species had a higher metabolic potential. And we think in this case, this is some sort of sign of higher metabolic potential being beneficial for the high altitude species to cope with these shorter activity periods, which are not really so much restrictive because these altitudes that we are talking to are around up to 1,500 or 1,000, right? So nothing extreme, but just a mild, shorter, colder environment in the altitude. And now let's move to the second example. This is a Galatia galoti lizard that live in the Tenerife Island in Canary Archipelago. This is the volcano Teide, which is the highest peak of Spain. It's a volcano that is in the Tenerife Island. And here we actually have an ongoing project that is run by PI Miguel Carretera and co-PI Caterina Pino, and I'm involved in. We also have a PhD student, Nina Seren, and other colleagues working together with us. We did several expeditions already to the Tenerife Island, and here we have this very, very cool lizard, Galotia galoti. Why cool? Because it's distributed in a huge altitudinal gradient. It is occurring across the island from 0 altitude to 3,700, the peak of volcano there on top there. And here you can see how this environmental gradient is allowing us to sample in a very, very different set of environmental conditions. And since we are sampling the same species in these very different temperatures, also a very different humidity range, there is change of other factors, we have this natural experiment going on. So we are already looking whether these traits, in my case, I'm investigating metabolism, if these traits are showing some signs of local adaptation to these environmental conditions. So temperature gradient is really high. There is the coldest, very cold top. There is even snow large part of the year there, so it's very restrictive for lizards. And then also this is very restrictive in very, very low altitude in the south, in this southern part of the island, because it's very dry and very hot in July. Then we have humidity. In humidity, it's actually considered, the top is considered a stone desert. It's very dry. And then very dry is also at very, very low altitudes. This is also very restrictive to lizards. And then, of course, the UV is stronger going upwards in elevation. And when we go above 1,500, these values of UV can be quite high, and they are producing oxidative stress in organisms if they are not able to cope with it. So we sampled in these 5 different altitudes, these 5 different environmental conditions, also the metabolism. And these are the results. We haven't yet published this, these are preliminary results. Now we also have some more population sampled, but here I'm just presenting these 5 populations that I showed you at these 5 elevations. So what we found actually, we found there are differences between populations. Now let's look at it maybe like this, it's going to be easier. So where did you, in this case, where did you find the highest metabolic potential? It wasn't on the top where it's the coldest, but here we found them, we found it actually here at 600 meters, whereas we found the lowest metabolic potential here on the top where it's the coldest, and here on the bottom where it's dry and hot. So this is now maybe a bit confusing, because we said before that the previous results showed us that having a colder environment would be better to have a higher metabolic potential. But I already mentioned there that's not very restrictive, right? Those environmental gradients that we were talking before, they are not that restrictive. Whereas here, where we have these huge altitudes on top, there it's really cold, very dry, and very high UV. So it's very stressful. And on the very, very bottom, it's very hot, very dry. UV is not a factor, but hot and dry is also very restrictive. And here is where we found the lowest metabolic potential, whereas we found the highest metabolic potential in a very rather good conditions for lizards, warm, humid, low UV. And what are these results telling us? First of all, they are telling us that metabolic potential, so the enzymatic capacity, changes across these different environmental conditions. So this is actually giving us a sign of some sort of local adaptation in these traits. But then we also got this signal of humidity being important in the environment. So very dry, really extremely dry conditions, they both gave a sign of low metabolic activity, and also high UV gave this signal. And now we're gonna see in the next example, I will explain why this is actually happening probably in this case. So I went to Denmark to do a short research stay there, and I worked there with this so no lizards, this is worms. So I worked with another ectotherm. I worked with a soil lizard, you can see it here on the picture, Enchytreus albidus. These are very small soil worms. They live in this coastal wet decaying seaweed, and they have this very large distribution across the latitudes. So this institute at Aarhus University that I visited, they already had samples of these soil worms that they collected. You see here from very far north, from Greenland and under very, very cold conditions, and then down to Spain. So this is all the same species. And they kept them there acclimatized at 5 degrees. And they already know quite a bunch about these soil worms. They knew that they are withstanding freezing. So they have these freeze tolerance abilities. But also freezing, we know that it's actually producing oxidative stress in organisms. So here we had the samples that are also across very different environmental range. And we have worms that are having a different ability to, a different tolerance to freezing, yeah? And in this sample, we looked at, again, at metabolism. So we were actually interested in this, whether this freezing as stressing factor, so the one that is producing oxidative stress, which freezing can be the same as UV or any other stressor that is producing oxidative stress. Does it have a link together with the metabolism? Does it show some sort of pattern? And we found it. So we found that worms that were coming from the northernmost parts and are the most freeze tolerant, they can even withstand, you see, minus 25 degrees, but they have a lot of oxidative stress. So these ones had the lowest metabolic potential, whereas the southern had the highest metabolic activity. So here again, with the temperature, we have together with another factor a sign that when the stress is high, so when the environments are very restrictive and there is some sort of stressor that is causing oxidative stress, we find this signal of low metabolic activity. Why is this? Why is that? Well, actually, it's like this. So the metabolism, when it's occurring, when metabolic reactions are occurring in our body, it is a normal byproduct are reactive oxygen species, which are causing oxidative stress to our cells. And then our body already has this so-called antioxidative mechanisms that will eliminate these radicals in order that our cells don't get damaged. But now if you imagine that your body has a lot of metabolism and a high potential for metabolism, then it's also having these high antioxidative mechanisms going on. And if there is another stressor that is causing a lot of oxidative stress present, these antioxidative mechanisms are already fully occupied with your high metabolism. They will not be able to kill, let's say, or to eliminate the radicals that are caused by another stressor. So apparently it looks like that the metabolism already in these very restrictive and stressful conditions, such as very cold, maybe high UV, and of course there could be other stressors like this, the metabolism is already low. The organisms develop a lower metabolic activity potential just in order to be able to have to cope with radicals that would occur under stress. So we think that in these worms, the adaptation of metabolism is to escape this freezing stress due to oxidative stress, to have a lower metabolic capacity per se. And this is probably also a reaction that we observed in Galatia galoti lizard, that from very, very restrictive environments, we have a lower metabolic activity to escape this additive effect of stress, oxidative stress from both metabolism and from the stressor that is coming from the environment. In that case, it was UV on top and dryness, so really low humidity, very dry conditions that are stressful to the lizards. And in the last example, I will go to another ectotherm. These are butterflies. And here I also did another short stay visiting Greifswald University in Germany. They had a response project they were looking at different traits in butterfly species, Pieris snappy. This species is also really widespread. And they were actually in this setup, in this project, they were using climate chambers. So they had an ongoing climate simulation experiment to which I joined, and then I measured metabolism. And actually they had 3 replicas of populations from Sweden, from Germany, and from Italy, where they collected females of this species of butterfly. They brought them back to the lab. They put them in the climate chambers on their this optimal temperature for development, which was around 26. The eggs were developed. They put the eggs on 26 until they developed until larvae. And then they had this experiment set up where they randomly placed butterflies from all 3 populations to either go on 18 degrees, and 18 degrees is the temperature in Sweden in July, where usually the larvae would develop, or to 26, which is the July temperature, average temperature of Italy, where the butterflies would develop usually. And they allowed them to develop until adults. And when they were adult, actually, then we took samples and we measured the metabolic activity as well. Well, actually, in this project, I just want to show you there were a lot of traits that were involved in this study. So morphological traits, performance traits, also times of development. But also, I did this part, I included the part on metabolic activity. And what we find out for metabolism, I will just present for metabolism here, is this. So the temperature effect. Yes, we found that Swedish butterflies that were that develop in nature, they develop at 18 degrees, and also here in the lab when they developed under 18 degrees, they had a higher potential metabolic activity than Italian butterflies that usually develop at 26. So that were developed at 26, they had a lower potential. And this is again similar to the metabolic syndrome that we were talking about in ectotherms, when it goes, when we are talking about these gradients that are not very, very restrictive, right? This is just a normal temperature range to which this species is clearly adapted to. But in cooler environments, they would have higher metabolic activity, and in warmer climates, they would have a lower metabolic activity. And this is probably this adaptation of metabolism to colder condition and these shorter activity periods, because of course in Sweden, again, the activity period is shorter, so they come out, butterflies can start flying later in the season and they retreat sooner than compared to Italy. And so therefore it's beneficial for them to have this higher metabolic activity, so they can, in a shorter activity season, do everything, the development of eggs and everything faster. But on the other hand, the second result that was even more interesting here now is something that we found something new. So when we put all 3 populations to 18 degrees, all 3 had at the end of the experiment a higher metabolic potential. So not only Swedish ones, and the same happened when we put all 3 butterflies at a high temperature set. All butterflies showed a low potential metabolic activity. And this is then a sign of plasticity as well. So apparently also already on the level of the enzymatic capacity of metabolism, we found a sign of plasticity. So if they are exposed to different temperatures, the metabolism is able to already respond in some sort of plastic manner. So with this, I will now finish my talk, and I hope I gave you some examples that clearly somehow showed you that we still need to work on this. There is still quite some open questions we have about metabolism, but what we already found with several studies is that there is definitely some sort of sign of adaptation and plasticity in metabolic traits on the level of enzymes and enzymatic capacity. But we also have to always take into account when we are studying lizards or ectotherms that use behavioral thermoregulation, we need to also know their preferred body temperature ranges. We need to know their behavioral responses to be able to clearly understand also the responses of metabolism and how these animals cope with everything. And not only temperature, we've seen also water losses, or water availability in the environment, dry conditions. This is very restrictive apparently to lizards. Then we found there are additive effects. So additive effects of multiple stressors in the environment that will also influence the response of metabolism and the actual traits of metabolism, because the oxidative stress is additive together with metabolism and other stress factors. So we need to know that, and we need to see, we found out there is actually a response to the oxidative stress on the level of metabolism. With this, I will say also thank you for your attention, and thanks. I really need to thank many people that I work with for their super enthusiastic collaborations, and I just put them here for you to see that this kind of work is not done alone, but always together with a bunch of people working together. Thank you.